I'm Dr. Nathaniel Chin, and you're listening to Dementia Matters, a podcast about Alzheimer's disease. Dementia Matters is a production of the Wisconsin Alzheimer's Disease Research Center. Our goal is to educate listeners on the latest news in Alzheimer's disease research and caregiver strategies. Thanks for joining us. Before today's episode, a quick disclaimer. The statements from Drs. Nathaniel Chin, Cynthia Carlsson, and Sterling Johnson do not reflect the opinions of the University of Wisconsin-Madison, UW Health, or the Veteran Affairs Healthcare System. The University of Wisconsin-Madison is one of 100 locations worldwide that are a part of the AHEAD study, and one of approximately 63 sites across North America that are a part of the A4 study. Both are clinical studies of the investigational treatment Lecanemab in people who may be at risk for memory problems. Dr. Carlsson serves as the principal investigator for the AHEAD and A4 studies at UW-Madison, and Drs. Chin and Johnson serve on the study teams. The AHEAD study is funded by the National Institute of Health, NIH, in partnership with the pharmaceutical company Eisai, and is being conducted by the NIH-funded Alzheimer's Clinical Trial Consortium, known as ACTC. The A4 study is funded by the National Institute on Aging, NIA, NIH, Eli Lilly and Company, and several philanthropic organizations. Read more about the AHEAD and A4 studies in the episode description. Dementia Matters and Drs. Chin, Carlsson, and Johnson receive funding from the National Institute on Aging and the National Institute of Health. Dr. Johnson has served on advisory boards for Roche Diagnostics, Prothena, Merck, and Eisai in the past 2 years. He receives research funding to the University of Wisconsin from Cerevu Technologies. Welcome back to Dementia Matters. On today's episode, I'm following up on a conversation I started back on January 17th regarding the FDA accelerated approval of the medication Lecanemab. For those who have not listened to that episode, I encourage you to do so, as we'll be going more in depth on this medication now, particularly the results of the Clarity AD clinical trial, which featured Lecanemab. While the FDA accelerated approval was not based on the results we'll be discussing today, the findings are critical in helping the scientific field, the FDA, and Medicare determine if this medication is clinically meaningful, a phrase I think you will be hearing a lot of in the future. Joining me today to discuss this clinical trial and its new drug are two returning guests, Dr. Cynthia Carlsson and Sterling Johnson. Dr. Carlsson is the director of the Wisconsin Alzheimer's Institute, as well as a professor, geriatrician, and Alzheimer's disease researcher at the University of Wisconsin School of Medicine and Public Health. Dr. Johnson is a professor, clinical neuropsychologist, leader of the Wisconsin Registry for Alzheimer's Prevention Study, known as WRAP, and the associate director of the Wisconsin Alzheimer's Institute. So thank you both for joining me on Dementia Matters.
Thanks, Nate. It's good to be here.
Thank you, Dr. Chin.
Oh, Cindy, you can call me Nate.
Yeah, thank you, Nate.
So Sterling, let's talk about the results of Clarity AD. So for our listeners, there's one primary outcome or endpoint, and then there are multiple secondary outcomes. But the primary outcome is an important one, a very important one, improving efficacy of a drug, and the one that's often talked about, although I would argue the secondary outcomes are very important too. So what was the primary outcome, Sterling, and what was this result?
Thanks, Nate. For this study, they chose the clinical dementia rating as their primary outcome. And there's a particular measure from this called the sum of boxes, which is just, as it sounds, it's adding up the points on this rating and reporting out those points. And when they did that, they showed that the treatment group on Lecanemab slowed down their rate of change on this on their score on this clinical dementia rating sum of boxes. They slowed down by 27% versus the placebo group that did not get the therapy.
So Cindy, can you briefly explain then this concept, this tool, the sum of boxes, and tell us what is meant? Now Sterling mentioned 27% reduced decline. The number 0.452 is also mentioned as a part of the score. Can you explain that to us?
Yeah, so with these studies, what they try to do is to get a kind of a full picture of the person's function. So this clinical dementia rating scale looks at 6 different domains. So it looks at memory, orientation, their judgment, how well they're able to problem solve, how they function in community affairs, how they function at home and in their hobbies and in their personal care. So there's different areas. They ask the participant or the patient and the family member or caregiver or someone who knows the person well, how well they're doing in these. And it's a very standardized approach, and then trying to see how well people are functioning in these different areas. And so with the lecanemab therapy, again, this sum of boxes improved. So kind of the sum of all these kind of added up improved by about 0.5. And so that reduction, again, there's people are questioning, is it that clinically meaningful? But again, some of the ratings, so to go from mild impairment to questionable impairment, or to go from moderate impairment to benign forgetfulness, can be within that range. So again, I think each person's different, how they would interpret how clinically meaningful this was. But for some people, it may be clinically meaningful. So, it could be clinically meaningful to them or to their family members. Both groups are still declining because they do still have Alzheimer's disease. The drug doesn't stop the disease, but it slows down the progression.
And thank you for emphasizing that last point, too, just for our listeners. This is not a cure for Alzheimer's disease, and people are still progressing, but they are noticing less change based on being on this medication versus this placebo. And so that was the primary outcome. So, Sterling, one of the secondary outcomes dealt with amyloid PET scans. And so, could you tell our listeners, what were they looking for, and was it significant what they were finding?
Yeah, this is a great question. Well, this drug is an anti-amyloid kind of a drug, and its target is the amyloid plaques in the brain. So the experiment looked at amyloid in the brain, and the way they could do that was with PET scans. And these are really a fancy way of doing imaging that is very specific to imaging the amyloid plaques in the brain, if in fact amyloid is there. And that was an entry criteria to being in the study. So we know that everybody had amyloid in their brain who was in this study. And the experimenters used this scale called the centiloid scale, and it's kind of a play on words of the centigrade temperature scale, which goes from 0 to 100. For 0 being where water is frozen and 100 being where water boils. Those are the reference points. And for the centiloid scale on this amyloid PET scan, 0 means you don't have any evidence of amyloid, and 100 means you have a level of amyloid that's very much like others who have dementia due to Alzheimer's disease. And in this study, the average centiloid was about 75, which is about 3/4 of the way there to being an AD-like level of amyloid plaques in the brain. And what the study did was by targeting amyloid, it lowered the amount of plaques in the brain from this starting point on average of 75, 59 points down to an average of about 18. And this was over a period of 18 months.
And so what is an abnormal then? I mean, you've said 100 is an abnormal centiloid. That's what someone with dementia with Alzheimer's disease. Is there a lower threshold, though, that one would consider, okay, now you're technically amyloid elevated? And then my second question to
Yeah.
you is, in removing amyloid, do you have to get to that level, do you think?
Well, the group level data showed that nearly everybody declined who was on the treatment, and in fact, as a group, they declined by 59 points from 75 down to 18. But what was intriguing was this particular study used a threshold of 30 as their cut point, or their threshold, so to speak, of being amyloid positive. And after 18 months on the drug, it seems like not everybody, but a good chunk of these people were below that level of 30 centiloids. At 1 year, it was 54. And I don't recall what the number was at the end of the trial, but it was a substantial number of, it was 68% actually became normal based on getting below this 30 centiloid level. And so that's pretty remarkable. Most people were reverted to normal by this drug.
And so, Cindy, in addition to an amyloid PET scan, there were other really important secondary outcomes. Can you go over a few of these and what the results were?
Yeah, so again, the study really tries to look at a variety of factors. So they look at things that are important to see if the drug's working. So things like amyloid levels in the brain. And they look at things like, does it help them think better? So looking at cognitive outcomes. And then the fuller picture, does it make a difference on their quality of life, how well they're functioning? So things that we really care about as people who may be living with this disease. So with the, again, the primary outcome was this clinical dementia rating scale, but they also had other measures. So another measure was the Alzheimer's disease composite score that looked at cognition and how it relates to function. And there's another activities of daily living scale too that looked at more functional outcomes. So there are a variety of measures that showed that not only did it, again, improve how much amyloid there was in the brain, but also it improved their daily function. It also improved their thinking abilities across a variety of types of thinking abilities. So that's partly what made this study more exciting, is that there was consistency across these things instead of just saying the one measure of amyloid in the brain improves without any impact on the person. This study showed that there was consistent improvement in a lot of these measures.
And so, Cindy, I'm going to ask you a tough question, but as a physician in a memory clinic who treats people living with thinking changes and who works with many other disciplines in doing so, do you think your colleagues and the patients that you see, would they find these results clinically meaningful?
I think there are some patients who would find this clinically meaningful. I think they're really want to protect their cognition, protect it from declining at all costs. And so I think there's other decision making that's going to come into play. So the clinical meaningfulness and also how the person views their time. So do they want to spend every 2 weeks coming in to get a therapy? Some patients don't want to see their doctor every 3 months because it's too much time taken away from gardening or fishing or other things they're enjoying. And so I think it's going to not only be a matter of do they think it's clinically meaningful, but is the benefit enough to counteract the time it's going to take them to have the infusions done. I think some clinicians say it's not very clinically meaningful. Others say we don't have anything that's better, and that they're encouraged. Some people are encouraged by the continued improvement over time for the duration of the study. So will it have even more effect after 18 months? Those are questions we don't know yet.
I appreciate your answer, because certainly risk benefit is something that matters a great deal. And I'm sure Medicare will be thinking about that too. But then also costs benefit, and cost doesn't have to be money. Cost can be time and the hassle factor. So it's not something I consider. So thank you for sharing that. There were other biomarker analyses that were done in addition to the amyloid PET scan. So Sterling, can you just provide a brief overview of some of the other key findings from the study?
Sure. Yeah. Our field really has several biomarkers now that are indicative of AD or indicative of having neuronal damage happening in the brain as a result of AD. And so this study looked at something called A beta 42 to 40, which is something you can get out of the spinal fluid or also out of the plasma. And it's an indicator of the amount of amyloid that might be present in those fluids. And that measure became more normal-like with treatment of lecanemab. And similarly, they measured something called p-tau 181, both in the spinal fluid and in plasma in the blood. And they found also with that it normalized. It became more normal-like, it was reduced with treatment, I should say. And then they also looked at another PET scan called a tau PET scan. And what they found with that is that the people on the drug had lower levels of tau, especially in the temporal lobe, which is the area that's most concentrated for tau proteins on these PET scans. And they looked at a few other things too, measures of neurodegeneration. One's called neurofilament light. One's called GFAP, which is a measure of astrocyte function. And there was finally one more called neurogranin, which is another measure of how well the neurons are functioning and how well the synapses are communicating. They found some mixed results with these. The neurofilament light measure didn't really change all that much. There was a slight trend that wasn't significant. The GFAP, which is this measure of inflammation-related change, improved significantly on lecanemab. Neurogranin, this measure of synapse function and how well neurons are communicating, that also improved. There was one other measure they did, which was MRI, and just looking at quantitatively at the amount of brain tissue before and after treatment. And what they found was a little bit curious. They found that there was slightly more atrophy in the group that had the lecanemab treatment. We don't know why that is. I think there'll be lots of research on this over the next coming months and years to help us understand why there would be this kind of paradoxical effect.
That's helpful for our listeners to understand if they weren't familiar with this particular clinical trial, but clinical trials in general, it's not just about amyloid protein or even tau protein, that there are other mechanisms or pathways of thinking change that these studies are looking at, inflammation being one of them. What does all that mean, though, when you think about all of these biomarkers either showing this expected result of change, reduction in change, or even trending in that way? What does that tell you about the amyloid cascade hypothesis or this ATN framework of amyloid tau and neurodegeneration?
I think when you look at the overall pattern here, the overall preponderance of the evidence, with the primary outcome being kind of clinical cognitive symptoms and the secondaries being other cognitive symptoms and activities of daily living, and the biomarker evidence, all of this is pointing to a drug that looks like it's working. It was most pronounced. The most pronounced effect looked like it was on amyloid, and that's I think that means that it's engaging the target. It's actually finding the amyloid plaque that's in the brain and reducing it. That seems to be the narrative that I'm seeing. And as a result of that, there's a reduction in symptoms. It's not complete, but at least there's a significant difference in symptoms and in these other biomarkers on this drug. And I think the preponderance and overall pattern here is that we have a favorable profile with this drug that looks like it's truly modifying the disease. It's not stopping it, but it's modifying it. And that's really, really incredible.
Now, Cindy, you hinted to this earlier in one of your answers, but there are researchers talking about potential cumulative effects of disease modifying therapy, just the way Sterling mentioned it. Now, what does that mean, though, a cumulative effect in a drug like lecanemab?
Well, these types of studies, they're short studies. Again, they're studying people over 18-month period of time. And obviously, many people with dementia due to Alzheimer's disease are going to be living beyond that. So trying to figure out what's going to happen beyond the 18-month period of time. And so sometimes we'll have additional information from what they call open label extension components of the studies, where people are able to get the true medication beyond the duration of the study. But sometimes they have to kind of guess what would happen if we kept the person on the study for 4 years, 5 years, et cetera. And so because of the way the graphs look and the results looked so far, it looked like people were continuing to have improvement in cognition and in these functions and their quality of life. So because, again, both groups are declining, but the group that's declining without the medication, it's declining at a faster rate. So again, if there's greater effect down the road, then, so for example, they're estimating that there may be a preservation of about 7 months of time of cognitive, stabilizing the cognitive function of someone. So, how clinically meaningful is that to have 7 more months of preserved cognitive function? So if that 7 months includes your granddaughter's, wedding or other big life events, that might be clinically meaningful for someone. They also, again, may progress to the next stage of dementia at a later point in time. So again, does that mean they can stay home longer, that their family can support them longer in their own home setting? So those things are sometimes really important questions for patients and families. We know that institutionalization, which is when someone moves from home to a nursing home or a higher level of care, it varies from person to person. So, if a patient happens to live next door to his daughter, who's a nurse, and his son's an EMT, and they've been in their neighborhood for 50 years and know everyone, that person might be able to stay in their home longer than somebody who maybe is new to their surroundings or doesn't have as many children nearby. So it's not just the effect of the drug on the person that's going to determine if someone moves to higher level care, but if the drug can help keep that person in their home setting, then that's valuable to patients, families, and economics as well, for the cost of providing care for people with dementia.
When you're leading me to my next question, which is that the study did look at other things such as well-being, quality of life. What did they find, Cindy?
Yeah, so they found that quality of life did seem to improve. Another thing which influences the care of the person with dementia is the care of the caregiver. So the person who's caring for that person, if they feel less stressed because the person's doing better, they're thinking better and able to engage in their own cares better, maybe they're sleeping better. So if the medicine helps with some of those aspects, then the caregiver may be sleeping better and functioning better. And so their what we call caregiver burden is less. And so they did some measures, a Zaret caregiver inventory scale that looks at some of those factors. So that seemed to improve as well. So a lot of different factors moving in the right direction, which is encouraging.
Sterling, can you tell our audience what subgroup analyses are, just in general, and then why they're done in a study?
Yeah, subgroup analysis is when you just look at a subset of people who are on the treatment or the placebo, and you look at those individuals to see if there was a difference there. And an example would be looking at older people versus younger people in the trial, or men versus women.
Well, and you kind of took my thunder with this question, though, Sterling, because the reason I'm bringing it up is that in the news, look, people are commenting that lecanemab may be more effective in older people versus younger people, and in men versus women. And from your scientific perspective, should we be looking at this cautiously, and why?
Well, these subgroups are really important for raising questions for further study. And we have a habit in our field of looking at subgroups because we've had, 2 decades of negative trials, and we've squinted at these little subgroups of certain demographics to see if there was any glimmer of hope in any of these subgroups. And so that's kind of been our habit as a field to try to learn from these failed trials over the years to see what the next trial might look like. In this case, the study was not really, they didn't plan on these things ahead of time. These are after the fact, and so there's not enough statistical firepower, if you will, to really have confidence in any of these subgroups. What they do is they, as I said before, they just raise questions for the next study, where we might want to look at men versus women with more intention on that in the future, or older versus younger subjects with more intention of balancing those groups and really having a proper study on age. But that's a question that we can now ask, and maybe the next study will answer it.
So instead of conclusions, these things should raise more important scientific questions.
Yes. Yeah.
So Cindy, you talked about risk benefit, cost benefit. So I'd like to talk a little bit about safety of the medication, particularly ARIA. And I did go into some ARIA in my introduction episode from January 17th. But I want to hear from you with a little bit more data that you have. How did lecanemab do in this regard?
So, right. So, again, people have raised the questions, rightly so, is that lecanemab does have side effects, and some of them are very serious. So, again, having good conversations about the side effects is really important for clinicians with the patients, and that's going to go into the decision making. So, ARIA are these amyloid-related imaging abnormalities that you've talked about before. And there's two types. There's one that's called hemorrhagic versus edema. So you have the H versus E. Hemorrhagic is more of the microhemorrhages, so tiny mini bleeds. And then the edema is just little bits of swelling in the brain. By calling them the I of the ARIA means that they're imaging. So they're things that we see on the brain scan. So not all of them does the person with these changes experience any symptoms with those. And so again, we have to distinguish them between what are we just seeing as kind of incidental findings on brain scans versus what are causing symptoms in the person that's going to affect their day-to-day life. So with the ARIA, they broke it down into the edema and the hemorrhage or the microhemorrhages, the mini bleeds. And what they found was that about 12.6% of people in the treatment group compared to the 1.7% in the placebo group, or the ones without active medication, had the ARIA that had the little edema with it, so the ARIA-E. But again, a very small percentage of those had symptoms. So it was only 2.8% of those who were on the treatment had ARIA-E with symptoms, and none of the participants on placebo did. So again, it's not very common unless you have the medication. But again, the hemorrhagic findings, they had about 17% of people who are on the treatment had the ARIA-H, and about 9% in the placebo group had the ARIA-H. So just to know that the amyloid itself in the blood vessels can lead to little microbleeds. So just having the disease in and of itself can lead to microbleeds. So that's why we're seeing some of these changes in the placebo group as well as the treatment group, but a higher percentage in the treatment groups. But again, many of these were not symptomatic either. So if you look down at the symptomatic, the people who had symptomatic of the microbleeds, it was extremely small. So like 0.7% in the treatment group and like almost 0.2% in the placebo group. So again, a lot of these factors have to be taken into context for, did they have edema, the ARIA-E with the ARIA-H? Were they just on the scan? Did the person have any symptoms with that? And kind of teasing apart a lot of those factors. The other thing that influences it is that people who have a genetic risk called apolipoprotein E epsilon 4 or ApoE4, something that increases our risk but doesn't necessarily mean for sure we'll get dementia. The ApoE4 carriers tended to have a greater risk of having some of the hemorrhages and the edema, so had the more side effects. So again, as people are coming into these trials, a lot of times for these studies, they're doing genetic testing to see, are they more prone to getting these side effects? And that'll probably become part of clinical practice, is that you may have to screen to see what their genetic risk factor is. You can decide what's your risk of having one of these microbleeds versus a microedema and it causing you symptoms where you would, have some clinical consequences.
It's a very unfortunate reality that the ApoE4 increases risk for having Alzheimer's disease, and that's the population we're trying to help with this drug, but then you're also at a higher risk of just having some of the side effects. So it does seem like an important piece of information for people potentially to know in the context of therapy, but it's good for us to be able to identify these things. Moving on from that, Cindy, I was hoping you could share with us how Clarity AD did as far as representation of the people who were actually in the study.
Yeah, so it's one thing that clinical trials across the board, but especially Alzheimer's disease clinical trials has not done a good job of, is making sure that the people who are included in the clinical trials are representative of those who are at risk for developing dementia due to Alzheimer's disease. So again, for a variety of reasons, probably a lot of social determinants of health, persons who are from African American descent or persons who are Hispanic, Latino background, and Native American have higher risks of developing dementia due to Alzheimer's, but yet they tend to be very underrepresented in these clinical trials. So this study made a concerted effort. Again, they did the study internationally, so they had participants from North America, Europe, Asia. So, again, obviously they had the larger Asian population, but within North America, they were able to increase the number of Hispanic participants and slightly increased the number of African Americans. So they had about 12% who were Hispanic. It was about 2 to 3% who were African American, and then the larger Asian population, chiefly from the cohort in Asia. So again, the representation was improved, but still not where we'd liked it to be, because we want to make sure that people from different backgrounds who have different constellation of risk factors, vascular risk factors, and other things are represented, so we can see, are these drugs safe and effective in these different kind of subgroups of individuals.
So we've talked about the results, the primary results, secondary outcomes. We've talked about the significance of those, as well as the potential side effects and the people being studied. So that's all being reviewed by the bodies that determine approval. And so, Sterling, what happens next? Where do we go from here?
Yeah, we're all waiting to know what's going to happen with these reviews, but it's in the FDA's hands, like you said, and they're reviewing the line item data and going over everything with fine detail, and they'll make their decision sometime in the late spring or summer of 2023. And then the Center for Medicaid Studies, that's the wrong word. What's the right word?
Services, right? Center for Medicare and Medicaid Services.
They will also be reviewing this probably after the FDA makes their decision. The Center for Medicare and Medicaid Studies will be reviewing this as well, and they'll be making decisions about whether this can be administered clinically and then whether it'll be reimbursed. It's an exciting time.
But a lot of waiting going on here, and so we'll make sure to provide updates as things happen. To end, Cindy, I'm going to ask you another hard question, and that is, let's say everything moves forward, the approvals happen. What actually needs to happen within healthcare in order for this medication to reach a patient?
That's a great question, and a lot of discussions happening around this, because, right now, dementia is underdiagnosed in our country. There's estimates that about 50 to 60% of people with dementia are diagnosed. Biomarkers are not widespread use, do not have widespread use in clinical practice. So a lot of clinicians are not comfortable using biomarkers, so using these amyloid PET scans or CSF levels or blood tests to decide if someone has elevated amyloid, to see if they're eligible to get the study drug. And then, so from there, again, improving diagnoses, improving understanding how to use biomarkers. And then from there, the clinicians have to understand, who's a good candidate for this. So what is their ApoE genotyping? Do they have other risks for cerebral hemorrhages? There's questions about whether someone on anticoagulation should get this medication. Then they have to be able to have enough infusion centers for people to come in every 2 weeks. The infusions take about an hour, but there's some prep time before and after, and some monitoring for allergic reactions. So again, there's that time. There's also careful monitoring that has to be done. So people have to be able to have an MRI scan before they have the infusions done, and also for monitoring afterwards. And so those MRI scans have to be done on a regular basis to make sure these ARIA changes, these microhemorrhages, microedemas, aren't occurring. We also need to have emergency room personnel who are comfortable when somebody comes in with a headache after getting an infusion, when to worry about a side effect versus just somebody having a headache. So there's a lot of training, infrastructure, follow-up that needs to happen. And this is happening in the context of, clinicians who aren't super comfortable many times managing dementia, caregivers who are already overburdened, and the patients who have memory loss. That's why they're being evaluated. So it adds extra layers of complexity to help kind of get this therapy into practice. And then again, in addition to healthcare systems having to decide if they're going to cover this expensive therapy and all the extra costs that come with administering it.
Well, with that said, so very exciting results, landscape-changing kind of study, but a lot still to understand, a lot still to determine.
Yes.
Well, I'd like to thank you both, Dr. Carlsson, Dr. Johnson, for joining me on Dementia Matters. I'm certain we'll be having more conversations as time progresses and we are learning more about this approval or not approval of lecanemab.
Thank you, Nate.
Thanks, Nate.
Thank you for listening to Dementia Matters. Follow us on Apple Podcasts, Spotify, Google Podcasts, or wherever you listen. Or tell your smart speaker to play the Dementia Matters podcast. Please rate us on your favorite podcast app. It helps other people find our show and lets us know how we're doing. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center at the University of Wisconsin-Madison. It receives funding from private, university, state, and national sources, including a grant from the National Institutes on Aging for Alzheimer's Disease Research Centers. This episode of Dementia Matters was produced by Amy Lambright Murphy and edited by Caitlin Rowerdink. Our musical jingle is Cases to Rest by Blue Dot Sessions. To learn more about the Wisconsin Alzheimer's Disease Research Center, check out our website at adrc.wisc.edu. That's adrc.wisc.edu. And follow us on Facebook and Twitter. If you have any questions or comments, please email us at dementiamatters@medicine.wisc.edu. Thanks for listening.